We will be going through several key systems in this course, from lung to kidney, neuromuscular, heart. All of those systems have the autonomic system influencing them. So I think it would do us well to consider broadly some principles on how the autonomic system works. So my task on this block is to do a kind of a whirlwind tour of the autonomic system. We know the autonomic system is made of the parasympathetic and sympathetic systems. So we will go through a broad sweep, and then we will loop back around as we work through each organ system and revisit the autonomic function, and in some cases go into a little more detail. So let's outline some principles regarding the autonomic system. This ANS, which is typically called, is controlling non-voluntary motor function in your body. So you don't have control over the parasympathetic or sympathetic systems in terms of dint of will. It's done below the level of consciousness. We call the part of the motor system that we do have voluntary control over the somatic nervous system. And that, of course, stands in contrast to the autonomic nervous system. And the somatic nervous system, again, deals with neurons coming from your motor cortices, and then they will go down, and then they will communicate with another neuron we call a motor neuron, and that neuron will leave the central nervous system, either the brainstem or the spinal cord, and will make its way to skeletal muscles. When it comes to the autonomics, the scheme is a little different. The autonomics, we have a communication with what we call a ganglion. A ganglion is a collection of neuronal cell bodies outside the central nervous system. If you have a collection of neuronal cell bodies within the central nervous system, and I'm defining the central nervous system as either brain, brainstem, or spinal cord, a collection of neuronal cell bodies within the central nervous system that has a similar function, we call that a nucleus. Now, nucleus is not in the sense of DNA, nucleus of a cell, but nucleus as in a collection of neuronal cell bodies that have like function. So again, collection of neuronal cell bodies within the central nervous system, call it a nucleus. If it's a collection of neuronal cell bodies outside the central nervous system, we call it a ganglion. That's singular. A ganglia is plural. So this is one of the differences between the autonomics and the somatic. Again, somatic, central nervous system, and this is either going to be the brain stem or the spinal cord. And of course, you have higher motor neurons. And let's just draw a little brain up here. There's my brain right there. Okay, so I'm going to have some motor neurons that will come down and will talk to other neurons either in the brain stem or the spinal cord, and then the brainstem or spinal cord. That's what this neuron here is symbolizing, and that neuron will send an axon out. It'll be a motor nerve, and it will then synapse onto a skeletal muscle. In the autonomic system, the scheme's a little different in the sense that we have, again, some neurons in the central nervous system, and we will learn in a bit where those actually are. They will then send an axon out leaving the central nervous system and progressing towards a ganglia. And this ganglia here is going to be synapsed upon, so it will release neurotransmitters onto this collection of neuronal cell bodies. And then those neuronal cell bodies will then send an axon out, forming a nerve, and they will then connect with or synapse on whatever their downstream target is. It could be smooth muscle, it could be cardiac muscle, it could be glands, etc. So again, it just depends upon what part of the autonomic system we're talking about. So this is a very good general contrasting scheme between somatic and autonomic. With the autonomics, it could either cause a contraction or it could cause a relaxation of smooth muscles. Again, it depends upon the system we're talking about, either the sympathetic or parasympathetic, or also could be talking about what organ systems are involved as well. And so we're going to find that it behaves differently. It depends upon, again, what part of the autonomics, and it depends upon the end organ system we're dealing with. So it could either cause, again, contraction of smooth muscles or a relaxation of smooth muscles. And we'll work on this as we go through. Another vocabulary that you're going to want to be aware of is that we have the term preganglionic fiber and postganglionic fiber. I believe you can figure out the pre and the post, and the pre and the post is referring to the ganglia here. So if we have two types of autonomic systems, parasympathetic and sympathetic, so we could have a parasympathetic preganglionic fiber, or we could have a parasympathetic postganglionic fiber, or we could have a sympathetic preganglionic fiber, or a sympathetic postganglionic fiber. So again, we have the para or the sympathetic para or the sympathetic that could be applied to preganglionic, postganglionic. A postganglionic fiber, by definition, is the axon. And let me say this as well, just to make sure we're clear. When I say fiber, in this context, I am speaking of axons from neurons. So we're not talking about the cereal All brand in terms of fibers. Okay, so we speak of fibers in terms of axons when we're dealing in this context here. So just make sure, I want to be clear with you on that. So if we're talking about a postganglionic fiber, then we're talking about the axon that is going to be leading to the end organ. If we're talking about a preganglionic fiber, then we're talking about an axon that is making its way to the neuronal cell body on which it will synapse. If you are confused regarding whether a fiber is preganglionic or postganglionic, all you have to do is ask the following question. And if you ask the question and answer the question correctly, you'll never get this mixed up. If I were to ask you, pointing to a fiber, is this preganglionic or postganglionic? The question you could ask is, where is the neuronal cell body from which that axon originates? If the answer is the cell body is in the peripheral nervous system living in a ganglia, then by definition it is a postganglionic fiber. Or if the answer is the neuron is residing in the central nervous system, spinal cord or brainstem, then the answer must be preganglionic fiber. So if you know where the neuron is from which the fiber is arising, the fiber in question, then you can answer the question correctly. So that's one way of looking at it. The highest control center for the autonomic system is the hypothalamus, which is in the center of your brain. So it is the hypothalamus that will be sending signals to this neuron here. So we're going to go hypothalamus. Okay, the neurons in the hypothalamus will talk to neurons here in the central nervous system, and then those neurons will then in turn send a fiber or axon that leaves the central nervous system to find its appropriate ganglia. Now, rest assured that the hypothalamus is not acting on its own. It has a lot of input from other neurons in the brain. The hypothalamus also can be affected by hormones and a lot of other biologically active molecules. So, again, a lot of influence on the hypothalamus. The hypothalamus then orchestrates the neurons in the central nervous system, and these neurons here are also either in the brainstem or the spinal cord. And they will then send their axons out, and then they will synapse onto a ganglia, and then those neurons will then talk to the end organ. Let's break down now the sympathetic and parasympathetic, and I'm going to be fairly general with you on this. As far as the sympathetic system is concerned, now, when I say sympathetic system, where you probably have heard of it in terms of fight or flight. So it is a system that gets you worked up. It is a system that increases your awareness. It gets ready for you to flee or to fight. My simple explanation of this, and this is what I use all the time explaining the sympathetic system, I call it the bear in the woods event. So you can imagine if you are walking in the mountains somewhere by yourself, alone, and you round a corner of a path, and you are face to face with a big old grizzly bear, eyeball to eyeball. And the grizzly bear is showing you his teeth. My guess is you are going to have an immediate physiological reaction. Your body will change. Your heart rate will go up. You will sweat profusely. You will breathe heavily. And of course, you will have a certain emotional valence associated with said events. Fear. So, what has happened is you processing the existential threat of the grizzly, you're going to have a neurological, physiological reaction to that. That is your sympathetic event. It's a bear in the woods event. That's what I call it. And so your body is getting ready to fight or flight. You're either going to flee from the bear or you're ready to fight the bear, depending upon how frisky you feel at the moment. But you're going to do one or the other. So why is your heart pounding? Well, it's pounding in anticipation of your skeletal muscles needing a lot of extra blood, glucose, oxygen. Why are you breathing heavily? Same thing. I'm going to need lots of oxygen. So whether I am running from the bear or fighting the bear, I'm going to need extra blood flow and extra oxygen. So why the sweating? Because in my effort to fight or flee, I'm going to be generating lots of heat, and I need to dissipate that heat. So again, the physiological response here has to do with preparing your body for intense physical activity. As far as the sympathetic system is concerned, you have a nucleus of neurons. Again, I'm using that word now, so you should know what that means. A nucleus of neurons meaning a collection of neurons somewhere within the central nervous system. And the one that is associated with the sympathetic system is called the intermediolateral cell column. And oftentimes you'll see it as the IML. This is actually a column. This nucleus is a column, and it lives in your spinal cord, from roughly level T1, T1 being thoracic 1. So you have spinal cord segments in your spinal cord, and they are roughly having to do with vertebra in your back. So you know that you have 12 thoracic vertebrae, and a thoracic vertebra is by definition attached to a rib, so you have 12 pairs of ribs. So your T1 is basically at the base of your neck, and you'll go down to T12, which is going to be about mid-back. So whenever your ribs run out, that's about where your thoracic vertebra runs out. And then, of course, caudal to your thoracic vertebra will be your lumbar vertebra. Going back up to your thoracic, rostral to your T1 vertebra will be your cervical vertebra. So you have numbered vertebra, and then you have numbered spinal cord segments that have something to do with the vertebra. They're not exactly lined up. In fact, the farther down you go in your spinal column, the less they are aligned, but they have something to do with each other. So that's close enough. So if I say I have a collection of neurons that runs from T1, that is thoracic level spinal cord 1, to L2, which is lumbar spinal cord segment L2, then that column of neurons are going to be the neurons that send their axons from the central nervous system to their external target. In this case, it would be a downstream ganglia. So we're talking about these guys right here for the sympathetics. These neurons here would be T1 through L2 in the spinal cord. Now, this is a bilateral structure, so you have a left one and you have a right one. And I have a cartoon on the next page that will help you with this. So those column of neurons, T1 through L2, are receiving input mostly from the hypothalamus. And of course, the hypothalamus is receiving lots of input from other systems. So they will come in, they will synapse, stimulate the IML. The IML will then send signals out, action potentials, to the neurons that leave the spinal cord and will talk to some other downstream ganglia. Here, you see. Now, I have some verbiage through here. It alludes to sympathetic chain ganglia, etc. That is not going to mean anything to you now. It will after I show you the drawing on the next page. So this paragraph, or the rest of this paragraph, will make sense or more sense to you when you see some cartoons. So I'm not going to burden you with that at present. The neurotransmitter that is used in the downstream ganglia, so now I'm talking about, let me clean this up a little bit. So I am talking about the neurotransmitter that's used here. And interestingly enough, whether it is the sympathetic system or the parasympathetic system, the same neurotransmitter is used. And the neurotransmitter that is used either in the sympathetic or the parasympathetic is acetylcholine. And we usually see this as ACH. Acetylcholine, very common neurotransmitter. Interestingly enough, it was the first neurotransmitter discovered back in the early 20th century. Oh, the 19-teens, somewhere along in there. There were two guys, scientists, last name Dale, and the other name is Lowy. they discovered acetylcholine and its function as a neurotransmitter. This was the first neurotransmitter. There was a long debate in science and medicine as to what was the nature of how electrical impulses were communicated from neuron to neuron or neuron to muscle. And the big debate was whether it was all electric or whether it involved a neurotransmitter or another molecule. And that debate raged for a fair while into the 20th century. Some people thought it was purely electrical. Others thought, no, you had to have an intermediate chemical or a molecule, which would be called a neurotransmitter, involved. So let me frame it this way. You know from high school or freshman biology that you have an axon coming out of a neuronal cell body, that the axon can have action potentials going down the axon. So how the signal is communicated from the beginning of the axon to the end of the axon, that's all electrical. So the question was, as one neuron talked to another neuron, was it somehow just a communication of that same electrical signal that just created an electrical signal in the downstream organ without any other chemical involved? Or was there release of a chemical that then in turn caused the next electrical impulse in the downstream nerve or neuron? That was the big debate. Is it electrical or is it chemical? Well, this was one of the first studies that suggested that it was probably involving another molecule, that it was not all electrical. So one of the first experiments on this, and it was done by Lowy, he was working with the vagus nerve, which you may already know and will know is a large nerve in the parasympathetic system. And its neurotransmitter, as we will about to see in a moment, is also acetylcholine. So they knew acetylcholine existed because Dale discovered its existence early on. And so what Lowy was doing was experimenting with the vagus nerve and a turtle heart. And he discovered that if you stimulate the vagus nerve, it slows the turtle heart down. And we'll find out in a bit why. So the vagus stimulation of the heart slows the heart rate down. So he did that experiment repeatedly, and again, stimulate the vagus nerve, go into the heart slows down, easy enough. So what he did was something rather clever. He took the fluid that was in the vicinity of the heart. Now the heart was taken from the turtle itself, so it's isolated, but still around some fluid so that it can maintain its metabolic integrity. It still can function for a little while at least. So and as you also know, the heart beats on its own. The heart does not need the central nervous system to beat. The heart has its own auto rhythm, and so the vagus nerve can influence the heart rate, but it certainly does not cause the heart rate. So what he did, he had two turtle hearts, again trying to figure out, is this purely electrical or is it chemical? So he stimulated the heart of the turtle, again it was removed, and then took the fluid around that heart that had slowed up in response to the vagus nerve, and then applied that fluid to another beating turtle heart that was not innervated by the vagus nerve. And when he did it, that heart also slowed down. So that was the first hint that something had to have been released, in this case acetylcholine, on the heart, and that you could take the acetylcholine and apply it to another beating turtle heart, and it would in fact slow it down. So that was suggestive that the nature of neuronal signaling had to do with chemicals as opposed to pure electrical impulses. So that was big stuff in the day, and Dale and Lowy actually won a Nobel Prize for that research. So a little bit of history on acetylcholine. Okay. Acetylcholine is not the only neurotransmitter involved. We're going to find out with other systems that norepinephrine and dopamine are also involved in this. So I think probably the best thing is to go to a diagram so that we can clarify this even further. And I'm going to draw your attention to these diagrams here to help us out. So again, we saw this earlier. That's my somatic. Okay, this is going to be, again, skeletal muscle. And what we find is acetylcholine is the neurotransmitter that these motor neurons, these are motor neurons, acetylcholine is actually released onto skeletal muscle for causing skeletal muscles to contract. So here we have the autonomics broken down to a parasympathetic and sympathetic. Both of them involve a ganglia. Ganglia, ganglia. And as I mentioned earlier, both of them release acetylcholine on that ganglia. Now, what happens is, now again, this is my pre-ganglionic sympathetic fiber. Post, well, I guess the P and that doesn't really work, does it? So let's just try this. Be a little more elaborate. This is my pre-ganglionic sympathetic fiber, and this would be my post-ganglionic sympathetic fiber. Okay, up here would be my pre-ganglionic parasympathetic fiber and my post-ganglionic parasympathetic fiber. So both the autonomics, para and sympathetic, both involve acetylcholine on the ganglia here, right? But when you go downstream, if we're talking the sympathetic, as far as the effector organ is concerned, one of the major ones is actually norepinephrine. It's not the only one, but it is the major neurotransmitter being used. Whereas on the parasympathetic, the acetylcholine is being released here on the effector organ as well. So while the neurotransmitters are the same in the ganglia for both sympathetic and parasympathetic, the neurotransmitter being used from the postganglionic fiber is different. In the parasympathetics, it's acetylcholine, and in sympathetics, it's norepinephrine. So this is the acetylcholine here that they were worried about or was looking at in terms of what was released on the turtle heart. Because if we're talking parasympathetics, which would be the vagus, of course, and in that case, the effector organ was the turtle heart. Okay, just to tie back our earlier illustration. Okay. Again, we'll return to parasympathetics in a moment. I do also want to share an aspect of the sympathetic system regarding the adrenal medulla. You know we have an adrenal gland, and its location is actually in the name, renal, okay, having to do with kidney. So your adrenal glands are sitting on the top of your kidneys, just happens to be there. And as you recall, perhaps from an earlier course, you have two different parts of the adrenal gland. You have the interior part of the adrenal gland, and that is called the adrenal medulla. And then you have an outer region, and that is called the adrenal cortex. And they're really two different glands that happen to be in the same place. Two completely different functions. So as far as the autonomics is concerned, we are focusing in on the adrenal medulla, the interior region. Of course, medulla means interior region. So it is within the medulla that you have neurons. And again, since we're talking sympathetic, there is my IML, my intermediate lateral cell column, T1 through L2. And so these neurons are here. Those neurons either go to a ganglia, or some of these neurons will go to the adrenal medulla. They will release acetylcholine. The nerves or the cells in the adrenal medulla, though, are not neurons. They're not ganglia like we have up here. So let me cartoon it out for you. Imagine I'm going to take these guys here, and they're going to be releasing their acetylcholine. The neurons in the medulla are just regular cells. And I have running through there, of course, blood vessels. I'm just going to go with that blood vessels there. The acetylcholine will bind to receptors on these cells and will stimulate these cells to release their contents, and their contents are going to be mostly epinephrine. The epinephrine is released, picked up by blood vessels, and distributed throughout the rest of the body. Those epinephrine molecules will then bind to receptors throughout the rest of the body, causing enhancement of function. Again, it depends upon the receptor, depends upon the organ. So you have two things kind of going on when you have a bear in the woods event. One is the system itself, the IML, and then the effector organ here. But the other is it's affecting your adrenal medulla. So I'm getting large amounts of norepinephrine on various end organs, but the adrenal medulla is releasing a hormone, epinephrine, and it is being carried throughout the body, also influencing. So you get kind of a double barrel shot of stimulation, so to speak. And the combinations of these allows you then to function in a way that is greatly enhanced. We've all heard the story of little old lady, grandma, 80 years old, walking her little dog, Fifi. And then you have a 4x4 rounding a corner, flips, crashes, and lands on little grandma's Fifi, little puppy. So now puppy is trapped under the 4x4 that is flipped. And grandma tosses her walker aside, runs to the 4x4, grabs it by the back bumper, lifts it up, and grabs her little precious Phoebe. And the dog is saved. How is that possible? Because in the moment of sympathetic stimulation through a variety of cellular processes, many of your functions are supercharged. Your muscles can do things that they normally could not do because of this surge of stimulating bioactive molecules called epinephrine and norepinephrine. To be sure, grandma will be quite sore the following day, but that is the power of sympathetic stimulation. complements of norepinephrine and epinephrine. So I think I will end this here, and we'll pick back up on this diagram in the next block.